This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to this exploration of our ancient mammalian ancestors. Today, we'll journey back over 200 million years to meet the Morganucodonts, tiny, shrew-like creatures that represent some of the earliest true mammals. We'll discover how these pioneers evolved remarkable Tribosphenic teeth, a revolutionary three-cusped design that allowed mammals to diversify their diets and ultimately dominate ecosystems worldwide. This innovation would prove essential to mammalian success, shaping everything from the smallest mouse to the largest whale, and yes, even us. Let's begin our story at the very dawn of the Age of Mammals. Chapter 1. Morganucodon Anatomy, Biology, Species Diversity, and Fossil Record. Welcome to our exploration of one of the most significant chapters in mammalian evolution. To understand how modern mammals came to dominate terrestrial ecosystems across the globe, we must journey back more than 200 million years to meet a tiny creature whose fossils tell an extraordinary story of transformation and adaptation. This animal, known as Morganucodon, represents a pivotal moment in evolutionary history when the fundamental characteristics that define mammals today were being assembled and refined through natural selection. The story of Morganucodon begins in the summer of 1947 in the rolling hills of southern Wales. Fieldwork was underway at Duchy Quarry in Glamorgan, where paleontologists were excavating gray conglomerate that had formed as ancient fissure fills within much older Carboniferous limestone. These fissures had acted as natural traps, preserving the remains of small animals that lived during the late Triassic and early Jurassic periods. 2 years later, in 1949, a German paleontologist named Walter Georg Kuhn was examining samples of this rock when he noticed something remarkable: a tiny lower cheek tooth of a primitive mammal, unlike anything previously described from European deposits. Kuhn named this new genus Morganucodon, deriving the name from Morganuk, the ancient name for South Glamorgan recorded in the Domesday Book. The species name, Watsoni, honored the distinguished paleontologist D.M.S. Watson. This initial discovery opened a floodgate of paleontological research. Throughout the 1950s, additional material surfaced from the same region. Kuhn himself described more remains in 1958 from Duchy Quarry, and in that same year, Kenneth Kermack and Francis Musset reported additional specimens from Pant Quarry, located about a mile away. These Welsh sites would prove to be extraordinarily productive, eventually yielding abundant and remarkably well-preserved material, though unfortunately, most specimens were disarticulated, making reconstruction of the complete animal a challenging puzzle. But Morganucodon was not confined to the misty valleys of Wales. In August of 1948, an expedition to Lufeng in Yunnan Province, China, yielded a 1-inch-long skull of what would later be recognized as a second species of Morganucodon. This precious specimen made an eventful journey from China to Beijing, then Peking, before eventually being deposited with Kenneth Kermack at University College London in 1960. Harold Rigney preliminarily described it in 1963, noting its striking similarity to the Welsh Morganucodon and naming it Morganucodon O'Leary, in honor of the Reverend Edgar O'Leary, who had originally collected the specimen. The discovery of Morganucodon in both Europe and Asia hinted at the widespread distribution of these early mammaliaforms during the Mesozoic era. The fossil record of Morganucodon would continue to grow. In 1978, the Chinese paleontologist C.C. Young described material from the Heiko-Peng locality near Lufeng, naming it Eozostrodon heikopengensis, based on an associated skull and dentary, along with additional jaw fragments. William Clemens, in a 1979 revision, assigned this species to Morganucodon based on its close similarity to the previously named species. The following year, Clemens described yet another species, Morganucodon peyeri, from isolated teeth found in late Triassic deposits near Hallau, Switzerland, naming it after the paleontologist Bernhard Peyer. This Swiss material represented some of the oldest known Morganucodon fossils, dating to the Rayetian stage of the late Triassic, roughly 205 million years ago. In 1981, Kermack, Musset, and Rigney published an extensive and detailed monograph on the skull of Morganucodon, synthesizing decades of research and providing the most comprehensive anatomical description to date. More recently, in 2016, Percy Butler and Denise Sigogno-Russell named Morganucodon tardus from an upper right molar collected from the Watton Cliff locality near Ype in Dorset, England. This specimen, dating to the late Bathonian stage of the Middle Jurassic, represented the youngest member of the genus. The species name tardus, Latin for late, acknowledged its position as the final representative of a lineage that had persisted for tens of millions of years. Today, we recognize 5 valid species of Morganucodon, spanning an impressive temporal range from the late Triassic through the Middle Jurassic, a duration of approximately 60 to 70 million years. This remarkable longevity speaks to the success of the Morganucodon body plan and ecological strategy. The geographic distribution is equally impressive, with fossils recovered from England, Wales, Switzerland, France, and China, suggesting these animals achieved widespread distribution across the northern continents during the early Mesozoic. So, what did Morganucodon actually look like? Reconstructing the appearance of an animal from fragmentary fossils requires careful analysis and comparison with living relatives. Based on skull and skeletal measurements, we can estimate that Morganucodon had a skull measuring 2 to 3 centimeters in length, with a presacral body length of approximately 10 centimeters, or roughly 4 inches. In general appearance, this animal would have resembled a modern shrew or mouse, with a small, compact body, relatively short limbs, and a moderately long tail. The animal was plantigrade, meaning it walked with its entire foot touching the ground, rather than on its toes, like many modern mammals. One of the most intriguing questions about Morganucodon concerns its body covering. Direct fossil evidence of fur is exceedingly rare in the paleontological record, but indirect evidence suggests Morganucodon was indeed furry. Researchers have identified specialized glands in the fossil material that are interpreted as grooming glands, structures that would only be necessary for an animal that needed to maintain a fur coat. This inference is supported by the small body size of Morganucodon and the probable need for insulation to maintain body temperature. Though, as we will discuss, the metabolism of these early mammaliforms differed significantly from that of modern mammals. The skeleton of Morganucodon exhibits a fascinating mosaic of primitive and derived characteristics. The atlas, the first cervical vertebra that supports the skull, shows unfused elements with a visible suture between the dens and the axis, a more primitive condition than seen in modern mammals. The cervical ribs were not fused to the vertebral centra, another primitive feature. The shoulder girdle retained both the coracoid and procoracoid bones, elements that are absent in modern placental and marsupial mammals, but present in monotremes and more primitive mammaliforms. The head of the humerus, the upper arm bone, was spherical as in modern mammals, but the spiral ulnar condyle retained a cynodont-like morphology, revealing the animal's evolutionary heritage. In the pelvic girdle, the pubis, ilium, and ischium remained unfused as separate bones throughout life. The skull of Morganucodon displays several features of particular evolutionary significance. The animal possessed a septomaxilla, a small bone in the snout region that represents a primitive character also found in earlier cynodonts. The orbitosphenoid, a bone forming part of the brain case, was fully ossified, and the anterior lamina was enlarged compared to more primitive forms. The squamosal, a bone that would become critically important in mammalian evolution, formed a narrow element positioned superficially to the petrosal and parietal bones. Significantly, Morganucodon possessed a larger cerebral capacity than its immediate predecessors, along with a longer cochlea in the inner ear, both features pointing toward the enhanced sensory capabilities that would become hallmarks of mammalian success. Perhaps the most revolutionary feature of Morganucodon was its dentition. Unlike the simple peg-like teeth of many reptiles, Morganucodon had evolved a heterodont dentition, meaning different types of teeth specialized for different functions. The tooth row included incisors at the front for grasping and nipping, followed by canines for puncturing, and then premolars and molars for processing food. This differentiation of tooth types represented a major advance in feeding efficiency. Moreover, Morganucodon was diphyodont, meaning it replaced its teeth just once during its lifetime, developing a set of milk teeth that were later replaced by permanent adult teeth. This pattern differs fundamentally from the polyphyodont condition, seen in most reptiles, where teeth are replaced continuously throughout life. The molars of Morganucodon show particularly sophisticated adaptations. These teeth featured a series of raised cusps and edges arranged in a linear pattern along the tooth row. When the upper and lower molars came together during chewing, these cusps and edges fit into corresponding valleys and spaces in the opposing teeth, creating an efficient cutting and shearing mechanism. Interestingly, the teeth did not fit perfectly when they first erupted. Instead, the precise occlusal relationship developed through wear as the animal used its teeth. This wear modified the shapes of the cusps and created matching surfaces that improved with use, a self-sharpening mechanism that maintained efficiency throughout the animal's life. The molars were specifically adapted for piercing and shear cutting, perfectly suited for processing tough insect prey. Evidence from the fossil sites where Morganucodon has been found provides valuable clues about its habitat and diet. Plant material from the conifer Hirmariella has been discovered in the same fissure fills that yielded Morganucodon fossils, indicating these animals lived in or near forested environments. The structure of the teeth, particularly the robust cusps and shearing edges, along with patterns of wear on fossil specimens, strongly suggests a diet focused on insects and other small animals. Modern analysis indicates a particular preference for hard prey, such as beetles, whose tough exoskeletons would have required precisely the kind of crushing and shearing capabilities that Morganucodon's dentition provided. The lifestyle of Morganucodon can be inferred through comparison with modern small mammals of similar size and presumed habits. Multiple lines of evidence point toward a nocturnal existence, with the animals spending daylight hours in a burrow. The evolution of nocturnality in the earliest mammals is supported by several considerations. First, small body size leads to rapid heat loss, making the cooler nighttime environment energetically challenging unless insulation like fur is present. Second, the enhanced sensory capabilities indicated by the enlarged brain and lengthened cochlea would have been advantageous for navigating and hunting in darkness. Third, nocturnality would have allowed early mammals to avoid competition and predation from the diverse array of diapsid reptiles, including early dinosaurs, that dominated daytime terrestrial ecosystems during the Triassic and Jurassic periods. This nocturnal bottleneck in early mammalian evolution would have profound consequences for the subsequent evolution of sensory systems, including the emphasis on hearing and smell over vision that characterizes many modern mammal lineages. Recent research has revolutionized our understanding of Morganucodon's physiology and life history. A 2020 study employing sophisticated analytical techniques examined growth patterns preserved in fossilized bone and teeth. The results proved surprising and challenged long-held assumptions about early mammalian biology. The study revealed that Morganucodon's metabolism was significantly slower than that of comparably sized modern mammals. Growth rates, while faster than typical reptiles, did not match the rapid development seen in living mammals. Even more remarkable, lifespan estimates derived from growth line analysis suggested the oldest known Morganucodon individual had lived for approximately 14 years, far longer than modern mammals of equivalent body size, which typically survive only 1 to 3 years in the wild. These findings indicate that Morganucodon had not yet evolved the fully endothermic, high metabolism physiology characteristic of living mammals. Instead, these animals occupied an intermediate physiological state, more active and metabolically capable than reptiles, but not yet achieving the metabolic intensity of modern mammals. The reproductive biology of Morganucodon remains largely inferential, but several lines of evidence provide intriguing hints. The structure and wear patterns of the teeth strongly suggest these animals practice lactation, nursing their young on milk produced by specialized mammary glands. The presence of a toothless stage during infancy, implied by the dipodont tooth replacement pattern, would have necessitated some form of parental provisioning, and lactation represents the most likely mechanism. The eggs of Morganucodon were probably small and leathery, similar to those of modern monotremes, the egg-laying mammals that represent the only surviving lineage retaining this primitive reproductive mode. The extended lifespan and relatively slow growth rate would have allowed for extended parental care, a pattern that may have contributed to the evolutionary success of early mammaliaforms by enhancing offspring survival. The jaw mechanism of Morganucodon reveals critical information about the evolutionary transition from cynodont ancestors to true mammals. One of the defining features of modern mammals is the dentary squamosal jaw joint, in which the lower jaw, composed of a single bone called the dentary, articulates directly with the squamosal bone of the skull. In contrast, reptiles and more primitive synapsids possess a jaw joint formed between the articular bone of the lower jaw and the quadrate bone of the skull. Remarkably, Morganucodon possessed both jaw joints simultaneously. The dentary had evolved a posterior process that contacted the squamosal, forming a new derived mammalian-type joint, positioned laterally. But Morganucodon had not yet abandoned the primitive articular quadrate joint, which persisted medially to the dentary squamosal articulation. This dual jaw joint system represents a transitional stage of profound evolutionary significance. The retention of the primitive jaw joint in Morganucodon is evidenced by several anatomical features visible in fossil specimens. The dentary possesses a trough along its internal surface near the posterior end called the Mammalian groove. During life, this groove housed Meckel's cartilage, a rod of cartilage that formed the core of the embryonic lower jaw. Additionally, the dentary shows distinct facets where other bones of the lower jaw attached, specifically the surangular and coronoid bones. These post-dentary bones, which are incorporated into the middle ear in modern mammals, remained part of the jaw apparatus in Morganucodon. The articular bone, which would eventually evolve into the malleus of the mammalian middle ear, still functioned as part of the jaw joint, articulating with the quadrate bone that would later become the incus. This dual jaw joint system raises fascinating questions about function and evolutionary advantage. Presumably, the new dentary squamosal joint was assuming an increasing proportion of the mechanical load during biting and chewing, while the old articular quadrate joint was gradually being relieved of load-bearing responsibilities. This mechanical unloading would have been a prerequisite for the eventual transformation of these jaw joint bones into delicate middle ear ossicles specialized for sound transmission. The process of this transformation and the selective pressures that drove it represents one of the most remarkable examples of evolutionary repurposing in the entire history of life, and we will explore this transition in detail in subsequent chapters. The genus Morganucodon and its close relatives demonstrate the enormous creativity of evolution during a pivotal period in vertebrate history. These small, superficially mouse-like animals were pioneers, experimenting with new anatomical configurations and physiological strategies that would eventually give rise to the extraordinary diversity of modern mammals. From the misty fissures of Jurassic Wales to the ancient river systems of Triassic China, Morganucodon and its kin scurried through the underbrush, crunching beetles and raising their young, steadily accumulating the adaptations that would allow their descendants to inherit the Earth. Chapter 2. The Dual Jaw Joint System. Among the most remarkable features preserved in the fossils of Morganucodon and its relatives is a skeletal innovation that illuminates one of evolution's most elegant transformations. This innovation is the dual jaw joint, a transitional anatomical configuration that simultaneously maintained an ancient reptilian heritage while pioneering a distinctly mammalian future. Understanding this dual articulation system is essential for comprehending how mammals evolved their characteristic jaw mechanics and feeding capabilities. The significance of the jaw joint might not be immediately obvious to those unfamiliar with vertebrate anatomy, but its evolutionary history represents one of the most thoroughly documented examples of structural transformation in the fossil record. In reptiles and their ancestors, the lower jaw articulates with the skull through a joint formed between two specific bones, the articular bone at the rear of the lower jaw and the quadrate bone of the skull. This articular quadrate joint had served vertebrates well for hundreds of millions of years, providing a sturdy hinge around which the jaw could rotate during feeding. However, in the lineage leading to mammals, a revolutionary change occurred. The dentary bone, which formed the tooth-bearing portion of the lower jaw, extended posteriorly and developed a new articulation point with the squamosal bone of the skull. This dentary squamosal joint would eventually become the exclusive jaw articulation in true mammals. Morganucodon stood precisely at the crossroads of this transition. Detailed examination of fossil specimens reveals unmistakable evidence that this animal possessed both jaw joints functioning simultaneously. The dentary had evolved a condylar process extending posteriorly to contact the squamosal bone laterally, creating the new mammalian-type joint. Yet the old articular quadrate connection persisted medially, positioned toward the inside of the skull. This dual articulation created a jaw mechanism fundamentally different from both its reptilian ancestors and its mammalian descendants. The presence of both joints raises intriguing biomechanical questions that lie at the heart of understanding this transitional system. How did these two articulations function together during feeding? Which joint bore the primary mechanical loads during biting? What happened during the power stroke when the jaws closed forcefully on prey? The answers reveal a sophisticated division of labor between the old and new joints. Biomechanical analysis of the dual jaw system suggests that the dentary squamosal joint had already assumed the primary load-bearing role in Morganucodon. The positioning of this joint closer to the tooth row provided superior mechanical advantage during biting. When the jaw closed, forces generated at the teeth traveled through the robust dentary bone directly to the skull via the squamosal articulation. This created an efficient load path that minimized bending stresses on the jaw. The condylar process of the dentary shows substantial bone thickness in fossil specimens, indicating it withstood considerable mechanical forces. The articulating surface with the squamosal is broad and well developed, further evidence of its load-bearing function. Meanwhile, the articular quadrate joint, though still present and functional, appears to have been progressively unloaded. The bones forming this ancient joint show reduced robustness compared to those of earlier cynodonts, where the articular quadrate joint bore the full mechanical stress of jaw operation. In Morganucodon, these bones had become more gracile, suggesting they were being relieved of their original biomechanical role. Rather than bearing primary loads, the articular quadrate joint likely functioned as a stabilizing hinge, guiding jaw motion and preventing lateral displacement. During the power stroke of a bite, the dentary squamosal joint handled compression forces, while the articular quadrate joint maintained alignment. This functional division created a mechanically robust system while simultaneously allowing evolutionary modification of the articular and quadrate bones. As these bones became freed from load-bearing responsibilities, they could be reduced in size and modified for other purposes without compromising feeding efficiency. Recent discoveries have illuminated the stages of this transition. In 2024, exceptionally preserved specimens from Jurassic deposits in China revealed intermediate conditions between the dual jaw joint system and the definitive mammalian middle ear, demonstrating that the transformation occurred through gradual, stepwise changes. These remarkable fossils and their implications for middle ear evolution will be explored in detail in the following chapter. The anatomical details preserved in Morganucodon fossils provide crucial insights into how the dual jaw system was structured. On the inner surface of the dentary, near its posterior end, a distinct groove runs along the length of the bone. This Mammalian groove housed Meckel's cartilage, a rod of embryonic cartilage that formed the core around which the lower jaw developed. In modern mammals, Meckel's cartilage breaks down during development, disappearing as the dentary ossifies. But in Morganucodon, Meckel's cartilage persisted into adulthood, maintaining a physical connection between the dentary and the postdentary bones throughout the animal's life. This persistent cartilaginous connection meant that movement of the jaw necessarily involved movement of the articular bone and the other postdentary elements. Adjacent to the Mammalian groove, the dentary of Morganucodon displays distinct facets where other bones attached. These facets mark the contact points for the cerangular and coronoid bones, elements that in reptiles formed substantial portions of the lower jaw, but in mammalian evolution became reduced or lost. The presence of these facets confirms that the postdentary bones remained integrated into the jaw structure, forming a composite lower jaw rather than existing as separate elements. The articular bone, positioned at the posterior terminus of this assembly, formed the medial jaw joint with the quadrate bone of the skull. The quadrate itself remained a fixed component of the skull, though its connection to the squamosal was becoming increasingly robust to accommodate the new lateral joint. The dual jaw joint system was not unique to Morganucodon. Fossil evidence indicates that this configuration was widespread among early mammalioforms during the late Triassic and early Jurassic periods. Close relatives of Morganucodon, including other members of the Morganucodonta, possessed similar dual articulations. So did members of other early mammaliaform groups, such as the Docodonts and Haramiyidans. This distribution suggests that the dual jaw joint represented a stable, functional configuration that persisted across multiple lineages for tens of millions of years. However, variation existed in the details of implementation. Some mammaliaforms retained more robust articular quadrate joints, suggesting continued mechanical participation in jaw function. Others showed more pronounced reduction of these bones, indicating more complete transfer of loads to the dentary squamosal articulation. The relative positions of the two joints also varied. In some species, the medial and lateral joints were nearly aligned in the antero-posterior axis, creating a broad articulation zone. In others, including Morganucodon, the dentary squamosal joint was positioned slightly anterior to the articular quadrate joint, creating a more complex rotational axis. These variations likely reflected different feeding behaviors and dietary specializations. Species that consumed harder prey items or employed more powerful bites may have maintained more robust articular quadrate joints to ensure jaw stability. Those feeding on softer foods could afford more complete reduction of the old joint. The mechanical demands of feeding thus shaped the expression of the dual jaw system across different lineages, producing a spectrum of transitional forms. The evolutionary pathway from the dual jaw joint to the single dentary squamosal joint of modern mammals involved profound changes in jaw musculature. In cynodonts and early mammaliaforms, multiple muscles attached to the postdentary bones, using them as leverage points for jaw movement. The pterygoideus muscle group, which in reptiles inserted primarily on the articular and angular bones, had to redirect its attachments as these bones reduced. Similarly, muscles controlling lateral jaw movements relied on the postdentary elements for their mechanical advantage. As the postdentary bones reduced in size and shifted toward different functions, the muscles associated with them either disappeared or redirected their attachments to the dentary. The masseter and temporalis muscles, which would become the primary jaw closers in modern mammals, expanded their origins and insertions, compensating for the loss of muscles associated with the postdentary elements. This muscular reorganization represented a major biomechanical restructuring. The enlarged temporalis muscle, originating on the side of the brain case and inserting on the coronoid process of the dentary, provided powerful jaw closing force directed through the dentary squamosal joint. The masseter muscle, extending from the zygomatic arch to the lateral surface of the dentary, added force from a different angle, improving the mechanical efficiency of the chewing stroke. The transition also required modifications to the sensory innervation of the jaw region. In reptiles, branches of the trigeminal nerve supply sensory information from the jaw joint, innervating the articular and quadrate bones. These nerves detect pressure, position, and movement, providing crucial feedback for controlling bite force and jaw position. As the dual jaw system evolved, both joints required appropriate innervation. The dentary squamosal joint developed its own sensory supply from the trigeminal nerve, while the articular quadrate joint retained its ancestral innervation. This dual innervation system allowed the animal to monitor both joints simultaneously, coordinating their function during feeding. The evolution of the definitive mammalian middle ear would eventually require the developmental breakdown of Meckel's cartilage, separating the middle ear ossicles from the jaw, a process whose molecular mechanisms have been elucidated through modern developmental studies. Another critical aspect of the dual jaw joint system involves the angle of the dentary. In modern mammals, the dentary possesses a distinct angular process extending posteriorly and ventrally from the jaw's posterior end. This angular process serves as an attachment site for the medial pterygoid muscle and helps define the shape of the jaw's posterior margin. Morganucodon lacked a true angular process. Instead, the posterior portion of the dentary showed only minimal inflection, remaining relatively straight along its ventral edge. This absence reflects the continued presence of the angular bone, one of the post-dentary elements inherited from cynodont ancestors. The angular bone occupied the position that would later be filled by the dentary's angular process. The development of the angular process in later mammalia forms coincided with the reduction and eventual loss of the angular bone. As the angular bone diminished, the dentary extended posteriorly and ventrally to replace it, eventually forming the characteristic angular process that defines the mammalian jaw. This replacement occurred gradually, with transitional forms showing both a reduced angular bone and an incipient angular process of the dentary. The functional result was similar, a projection providing leverage for jaw muscles, but the structural composition changed from a multi-bone assembly to a single enlarged dentary. The dual jaw joint system of Morganucodon and its relatives thus represents both an ending and a beginning. It marked the final stage of the ancient articular quadrate jaw joint, which had served synapsids since their origin in the Carboniferous period. Simultaneously, it established the foundation for the dentary squamosal joint that would characterize all subsequent mammalian evolution. This transitional configuration maintained full feeding functionality while creating the anatomical opportunity for revolutionary improvements in other systems. The gradual unloading of the articular quadrate joint freed these bones for modification, setting the stage for their eventual transformation into specialized auditory structures. Understanding the dual jaw joint system illuminates the incremental nature of major evolutionary transitions. Natural selection did not leap from one functional configuration to another in a single bound. Instead, it worked through viable intermediates, each representing a complete functional organism. Morganucodon could feed effectively with its dual jaw joint, crushing beetles and other prey with no loss of efficiency compared to its ancestors. Yet this very system contained the seeds of future transformation, demonstrating how evolution builds new adaptations from modified versions of existing structures, rather than creating them anew. Chapter 3. From Jaw Hinge to Hearing. The transformation of bones that once formed the hinge of the jaw into delicate sound-transmitting ossicles represents one of the most extraordinary examples of evolutionary repurposing in the entire history of vertebrate life. This transformation enabled mammals to achieve exceptional auditory sensitivity, opening new sensory worlds that contributed to their eventual success across terrestrial ecosystems. Understanding how this remarkable transition occurred required integrating evidence from comparative anatomy, paleontology, embryology, and developmental biology across nearly 2 centuries of scientific investigation. In 1837, a German anatomist named Karl Reichert proposed a revolutionary hypothesis based on careful comparative studies of vertebrate skull development. Reichert observed that the tiny bones of the mammalian middle ear, the malleus and incus, bore striking resemblances to the articular and quadrate bones that formed the jaw joint in reptiles and birds. This was not a superficial similarity, but extended to their embryonic origins, their relative positions, and their connections to surrounding structures. Reichert's proposal was audacious for its time, suggesting that bones performing entirely different functions in different animal groups were in fact the same structures, modified by evolution to serve new roles. Reichert's hypothesis gained substantial support in 1912 when Ernst Gaupp expanded the theory with detailed descriptions of skull development in various vertebrates. Gaupp documented the existence of what he termed a primary jaw joint between the malleus and incus, corresponding to the ancient articular quadrate articulation, and a secondary jaw joint between the dentary and squamosal bones that was unique to mammals. This dual articulation provided compelling evidence that the mammalian middle ear bones were indeed derived from the ancestral jaw joint. For many decades, debate continued about the precise mechanisms and sequence of this transformation, but recent fossil discoveries, particularly those examined in the previous chapter, have confirmed the gradual nature of this transition and the mechanical unloading that enabled it. The most powerful confirmation of the articular malleus and quadrate incus homology, however, comes not from fossils, but from embryology and developmental biology. When scientists examined the embryonic development of the mammalian skull, they discovered that the malleus and incus developed from exactly the same embryonic tissues that give rise to the articular and quadrate in reptiles and birds. Specifically, both sets of bones originate from neural crest cells of the first pharyngeal arch, the same embryonic structure that forms the mandibular segment of the jaw. This shared developmental origin provides unambiguous evidence of homology. Evolution does not create new structures from nothing, but repurposes existing developmental programs, and the malleus and incus retain the developmental signature of their origins as jawbones. Furthermore, the developmental sequence mirrors the evolutionary pathway revealed by fossils. In mammalian embryos, the malleus initially forms at the posterior end of Meckel's cartilage, the embryonic cartilage rod that forms the core of the developing lower jaw, in precisely the position where the articular bone forms in reptiles. As development proceeds, the malleus remains connected to Meckel's cartilage and to the developing dentary bone, recapitulating the ancestral conditions seen in fossils with dual jaw joints. Only later in development does the malleus separate from the jaw as Meckel's cartilage breaks down. This developmental sequence preserves a record of the evolutionary history, with the embryo passing through stages that correspond to ancestral adult configurations. The phenomenon of embryos recapitulating evolutionary history is not universal, but in the case of the mammalian middle ear, the correspondence is remarkably clear. The temporary connection between the malleus and the dentary in mammalian embryos has no function in modern mammals. It exists solely because the developmental program inherited from ancestors still creates this connection before later breaking it. This vestigial developmental stage would make no sense except as a legacy of evolutionary history, providing some of the strongest evidence for the transformation of jawbones into ear bones. The breakdown of Meckel's cartilage represents a crucial innovation that enabled the complete separation of the middle ear ossicles from the jaw. In reptiles and early mammalia forms, Meckel's cartilage persists throughout life, maintaining a physical connection between the jaw and the ear region. In modern mammals, however, most of Meckel's cartilage degrades during embryonic development through a precisely orchestrated molecular program. Research published in 2017 identified specific developmental mechanisms controlling this breakdown. The process involves complex signaling pathways that activate at particular developmental stages, causing portions of the cartilage to be resorbed while other portions are retained and ossified. This programmed degradation occurs in a specific sequence. The middle section of Meckel's cartilage, spanning the distance between the dentary and the malleus, breaks down first, physically separating the middle ear from the jaw. Small remnants of Meckel's cartilage persist associated with the malleus, forming the anterior process of the malleus and contributing to other structures in the middle ear region. The anterior-most portion of Meckel's cartilage ossifies to become the mental ossicles near the chin. The evolution of the molecular mechanisms controlling Meckel's cartilage breakdown was essential for establishing the definitive mammalian middle ear. These mechanisms can be experimentally manipulated in laboratory animals, and when scientists disrupt the signaling pathways responsible for cartilage resorption, the malleus fails to separate from the dentary, recreating an ancestral condition in modern mammals. Marsupials provide particularly illuminating evidence for middle ear evolution because they retain certain features that are more primitive than those of placental mammals. In marsupial development, the middle ear ossicles remain connected to the dentary somewhat longer than in placentals, and the breakdown of Meckel's cartilage occurs later in development, sometimes not being completed until after birth. Newborn opossums, for instance, can be observed with their middle ear ossicles still attached to their lower jaws, a condition they share with ancient mammalia forms, but which is only temporary in modern marsupials. This extended connection allows researchers to observe the separation process in greater detail and confirms that even in modern therians, the developmental pathway recapitulates aspects of the evolutionary transition visible in fossils. The marsupial pattern represents an intermediate developmental timing between the ancestral condition of permanent connection and the placental condition of early embryonic separation. The question naturally arises, what functional advantages drove this remarkable transformation? Why would natural selection favor the conversion of jaw bones into ear bones? The answer lies in the physics of sound transmission and the dramatic enhancement of auditory sensitivity that the three-ossicle system provides. In reptiles and birds, a single middle ear ossicle, the collemula or stapes, spans the air-filled middle ear cavity. This single bone transmits vibrations from the tympanic membrane to the inner ear, but it has significant limitations in efficiency and frequency range. The addition of two more ossicles, the malleus and incus, created a lever system that provided crucial mechanical advantage. The three bones work together as a coupled system, where the malleus receives vibrations from the tympanic membrane, transfers them to the incus through their articulation, and the incus, in turn, transfers them to the stapes, which delivers them to the inner ear. The geometry of this chain creates impedance matching between the air-filled outer and middle ear and the fluid-filled inner ear. Sound travels easily through air, but is largely reflected when it encounters the much denser fluid of the inner ear. Without impedance matching, about 99% of sound energy would be lost at this transition. The mammalian middle ear ossicles solve this problem by amplifying the force of vibrations while reducing their amplitude, concentrating the sound energy into the smaller area of the oval window where the stapes contacts the inner ear. Moreover, the three-ossicle system dramatically enhanced sensitivity to high-frequency sounds. The smaller, more delicate bones could respond to rapid vibrations more effectively than the larger, heavier single ossicle of reptiles. The mass and stiffness of the ossicles determine which frequencies they can transmit efficiently, and the miniaturized mammalian ossicles extended the upper range of hearing considerably. This improved high-frequency hearing would have been particularly advantageous for small nocturnal animals like early mammals, enabling them to detect the high-pitched sounds of insect prey moving through leaf litter, the ultrasonic vocalizations of conspecifics, or the approach of predators in darkness. The selective advantage of enhanced hearing in the nocturnal environment where early mammals lived provided strong evolutionary pressure favoring the refinement of the middle ear apparatus. Research using computational modeling has demonstrated that as body size decreased in early mammalia forms, the biomechanical demands on the jaw joint changed in ways that facilitated middle ear evolution. Smaller animals with smaller food items and lower absolute bite forces required less robust jaw joints. This mechanical scaling created an opportunity for the posterior jaw bones to become smaller and lighter without compromising feeding efficiency. Miniaturization was not merely a consequence of the middle ear transformation, but an enabling factor that facilitated it, creating conditions where bones could be repurposed for an auditory role while still maintaining jaw function during the transitional stages. The complete mammalian middle ear apparatus involves more than just the three ossicles. These bones are housed within a protected chamber, the auditory bulla, which shields them from damage and helps define the air-filled middle ear cavity essential for sound transmission. The formation of the bulla represents another crucial innovation in mammalian ear evolution, involving contributions from several different bones, in patterns that vary among different mammalian lineages. One important component is the tympanic ring, a delicate ring of bone that supports the tympanic membrane, or eardrum. The tympanic ring forms through a process of membranous ossification, with bone depositing directly in connective tissue rather than replacing a cartilage precursor. In mammalian development, the tympanic ring forms in close association with Meckel's cartilage and the developing malleus. Another small bone, called the gonial, ossifies between the tympanic ring and the malleus, serving as an investing bone for the malleus. The malleus is therefore a compound structure with a dual origin, partly from endochondral ossification of first arch cartilage and partly from the gonial bone that invades and fuses with it. This composite nature of the malleus reflects its complex evolutionary history. The configuration of the middle ear differs between the major mammalian groups, reflecting their independent evolutionary histories. In monotremes, the living egg-laying mammals, represented by the platypus and echidnas, the middle ear shows a unique arrangement. Adult monotremes lack a tympanic membrane and functional middle ear ossicles, appearing to hear primarily through bone conduction. However, young monotremes possess these structures, and fossil relatives of monotremes had well-developed middle ears. This suggests monotremes have secondarily lost functional middle ears, likely as adaptations to their specialized lifestyles, involving aquatic foraging or soil digging, where airborne sound transmission is less important. Despite this secondary loss, developmental studies of monotreme embryos confirm that their vestigial middle ear bones develop from the same embryonic origins as those of other mammals, supporting the common evolutionary origin of all mammalian middle ears from the ancestral jaw joint bones. In contrast, marsupials and placentals, collectively known as therians, possess fully functional three-ossicle middle ears with enclosed tympanic cavities. Within therians, the detailed anatomy of the bulla and the relationships of the ossicles vary. In some groups, the tympanic ring remains a separate element throughout life, while in others, it fuses with surrounding bones to form a composite bulla. These variations reflect the evolutionary plasticity of the middle ear region, even after the basic three-ossicle configuration was established. Recent discoveries have also revealed that the separation of the middle ear from the jaw occurred independently in different mammalian lineages at different times. A study published in 2020 described a Cretaceous stem therian from China in which the middle ear ossicles had achieved complete separation from the dentary, representing an integrated hearing module that was fully decoupled from the chewing apparatus. This specimen demonstrated that by the early Cretaceous, at least some mammalian lineages had achieved the definitive mammalian middle ear configuration. However, other lineages retained the ossicles attached to the jaw much later, indicating that the transition occurred at different rates in different groups. The fossil record has also documented variation in how the ossicles separated. In some lineages, the malleus appears to have separated first, breaking its connection to the dentary while remaining physically connected to the incus. In other cases, the entire ossicular chain separated as a unit. These different pathways toward the same end result illustrate that evolution does not follow a single predetermined route, but explores multiple solutions to the same functional challenge. One intriguing aspect of middle ear evolution concerns the fate of other postdentary bones beyond the articular and quadrate. In cynodonts and early mammalia forms, several bones contributed to the posterior portion of the lower jaw, including the angular, surangular, and coronoid. As the dentary expanded posteriorly and took over the entire jaw function, these bones were reduced or lost. The angular bone in some mammalian lineages contributed to the formation of the ectotympanic, part of the structure supporting the eardrum, thus finding a new role in the auditory apparatus, just as the articular and quadrate did. The surangular and coronoid simply disappeared, their developmental programs suppressed in the mammalian lineage. The transformation of the jaw joint into the middle ear fundamentally changed the constraints on skull evolution in mammals. With the middle ear ossicles freed from load-bearing duties, they could be modified to enhance hearing without concern for compromising feeding mechanics. Conversely, the jaw joint could be optimized purely for feeding efficiency without needing to serve dual functions. This decoupling of functions permitted greater evolutionary flexibility, contributing to the remarkable diversification of both jaw morphology and auditory capabilities among mammals. The same evolutionary transition that gave mammals their exceptional hearing also liberated their feeding apparatus to evolve in new directions, enabling the extraordinary dietary diversity seen among modern mammals. The evolution of the mammalian middle ear from jaw joint bones stands as one of the most thoroughly documented major transitions in vertebrate evolution. From Reichert's initial anatomical observations in the 19th century, through Gaup's developmental studies, to modern molecular and paleontological research, the evidence has converged on a coherent picture of how this transformation occurred. The story involves the gradual mechanical unloading of the ancestral jaw joint, miniaturization and refinement of the bones involved, eventual separation from the dentary through the breakdown of Meckel's cartilage, and enclosure within a protective bulla. Each step is documented in fossils, recapitulated in development, and explicable in terms of functional advantages for hearing. This transformation enabled mammals to achieve extraordinary auditory sensitivity, opening new sensory worlds and contributing to their evolutionary success. Chapter 4. The Tribosphenic Revolution. If the evolution of the mammalian middle ear represents an extraordinary repurposing of existing structures for a new function, the evolution of tribosphenic molars demonstrates nature's capacity to create entirely novel functional complexes through the modification and elaboration of simpler ancestral patterns. The tribosphenic molar would prove to be one of the most consequential dental innovations in vertebrate history, enabling the ancestors of modern marsupials and placentals to process food with unprecedented efficiency and versatility. Understanding this dental revolution requires us to examine both the intricate three-dimensional architecture of these teeth and the biomechanical principles that made them so remarkably successful. The term tribosphenic itself provides clues to the functional design of these molars. The word combines elements meaning three cusps and wedge or basin, referring to the characteristic arrangement of three primary cusps on the upper molar and the basin-like structure on the lower molar. But this simple description barely hints at the sophisticated engineering embodied in these teeth. When we examine tribosphenic molars in detail, we discover a complex system of interlocking cusps, cutting edges, and grinding surfaces that work together in precisely coordinated sequences during the chewing cycle. Let us begin with the upper tribosphenic molar, examining it as if we were looking down at the chewing surface from above. The most prominent feature is the trigon, a triangular arrangement of three major cusps. The protocone rises from the lingual side of the tooth, the side nearest the tongue. This cusp represents a genuine evolutionary novelty, absent in more primitive mammalian teeth. Its appearance marks a watershed moment in dental evolution, so significant that its presence defines membership in the Tribosphenida. The protocone functions primarily as a crushing structure, descending into a corresponding basin on the lower molar, like a pestle into a mortar. On the buccal side of the upper molar, toward the cheek, stand two additional major cusps. The pericone occupies the anterior position, while the metacone sits posteriorly. These cusps are evolutionarily ancient, traceable back through earlier mammaliform lineages, and ultimately to the simple conical teeth of more primitive synapsids. Between and around these primary cusps run a series of crests, sharp ridges of enamel that create cutting edges. The pre-pericrista extends from the pericone toward the front of the tooth, while the post-pericrista runs from the pericone toward the protocone. Similarly, the pre-metacrista and post-metacrista radiate from the metacone. These crests form a network of shearing surfaces that slice through food as the jaws close. Along the buccal margin of the upper molar extends a shelf-like structure called the stylar shelf. This platform bears several smaller accessory cusps, including the peristyle at the front corner and the metastyle at the rear corner. In some mammals, additional stylar cusps develop, designated by letters. Particularly important is stylar cusp C, positioned between the peristyle and metastyle. The presence and size of these stylar cusps varies considerably among different mammalian groups, reflecting diverse dietary specializations. Running along the stylar shelf, connecting these cusps, is a ridge called the ectoloph, which adds additional cutting capability to the tooth's buccal edge. Encircling the base of the protocone on the lingual side runs a ridge called the cingulum. This structure serves multiple functions, including strengthening the tooth base, creating additional grinding surfaces, and helping to distribute mechanical stresses away from the roots during heavy chewing. In many mammalian lineages, portions of the cingulum become enlarged and elaborate, sometimes developing into additional cusps that further enhance the tooth's functional versatility. Now, let us turn our attention to the lower tribosphenic molar, which exhibits an equally sophisticated but distinctly different architecture. The anterior portion of the lower molar is dominated by the trigonid, a group of three major cusps arranged in a triangle. The protoconid, the tallest of these cusps, rises from the buccal side. On the lingual side sits the metaconid, roughly equivalent in height to the protoconid and positioned slightly posteriorly. At the front of the trigonid stands the periconid, though this cusp shows a tendency toward reduction or loss in many mammalian lineages, particularly those emphasizing grinding over shearing. The trigonid functions primarily as a cutting and slicing structure. The three cusps are connected by sharp crests that form highly effective shearing blades. As the jaw closes and the lower molar moves past the upper molar, these crests slide against corresponding surfaces on the upper tooth, slicing through food with scissor-like precision. The trigonid is typically elevated above the posterior portion of the tooth, creating a differential height that enhances its cutting action during the initial phase of the chewing stroke. Posterior to the trigonid lies the most revolutionary feature of the tribosphenic lower molar, the talonide basin. This concave depression is bordered by three cusps, the hypoconid on the buccal side, the entoconid on the lingual side, and the smaller hypoconulid at the posterior margin. The talonide basin serves as the mortar into which the protocone of the upper molar descends, creating a grinding surface where food can be crushed and pulverized. The walls of the basin, formed by the surrounding cusps and their connecting crests, help to retain food particles during the grinding phase of mastication, ensuring thorough processing before swallowing. The functional beauty of the tribosphenic molar system becomes apparent when we observe how the upper and lower teeth interact during the chewing cycle. As the jaw begins to close, the trigonod of the lower molar approaches the space between two upper molars. The cutting crests of the trigonod align with corresponding surfaces on the upper teeth, and as jaw closure continues, these surfaces slide past each other in a shearing action that slices through food. This shearing phase is dominated by what researchers designate as wear facets 1 and 2, specific areas of tooth-to-tooth contact that develop characteristic patterns of wear over the life of the animal. As the jaw continues closing and shifts slightly lingually, the protocone of the upper molar enters the talonid basin of the lower molar. This initiates the crushing and grinding phase, characterized by wear facets 3 and 4. The protocone acts like a pestle, crushing food particles against the floor of the talonid basin. The walls of the basin help contain the food while grinding proceeds, preventing particles from escaping before they are adequately processed. Additional wear facets, designated 5 and 6, develop on other contact surfaces, creating a total of 6 matching shearing and crushing zones between upper and lower tribosphenic molars. The sophistication of this system extends beyond the simple closing of the jaws. During tribosphenic mastication, the lower jaw executes a complex rotational movement involving both vertical closure and mediolateral displacement. As the jaw closes, it simultaneously undergoes an eversion-inversion sequence, rolling the lower molars inward toward the midline of the mouth by approximately 10 degrees in many mammals. This rotational grinding stroke transforms the simple up and down motion of earlier mammalia forms into a true mortar and pestle action. The protocone sweeps across the talonid basin in a circular motion, grinding food particles against the basin floor and walls with far greater effectiveness than simple vertical compression could achieve. This dynamic three-dimensional movement represents a fundamental innovation in mammalian feeding mechanics, multiplying the food processing capacity of each chewing cycle. To appreciate the revolutionary nature of this system, we must compare it with simpler designs. As we saw in Chapter 1, early mammalia forms like Morganucodon possessed molars with 3 primary cusps arranged in a line, creating an effective cutting and shearing mechanism for processing insects. However, this linear arrangement had significant limitations. The cutting action was essentially two-dimensional, with minimal grinding capability, restricting dietary flexibility to foods that required primarily shearing, rather than both cutting and grinding. The transition from linear cusp patterns to the triangulated arrangement of tribosphenic molars involved a fundamental reorganization of molar architecture. The critical innovation was the movement of cusps from a single anterior-posterior line into a triangular configuration. In the upper molar, this meant rotating the metacone from a position directly behind the pericone to a position more lingually, creating the trigon triangle. Simultaneously, the protocone appeared as a new structure on the lingual side. In the lower molar, the metaconid shifted from a position in line with the protocone to a lingual position, while the talonid basin developed posteriorly as an entirely new structure. Remarkably, the developmental mechanisms underlying these complex dental structures are governed by relatively simple genetic programs. Cusps develop in sequence during tooth formation, with earlier forming cusps producing inhibitory signals that suppress the formation of additional cusps in their immediate vicinity. This inhibitory cascade model explains how cusp spacing and size are determined. The balance between activating signals that promote cusp formation and inhibiting signals that prevent it determines the final pattern. Earlier forming cusps are typically larger because they develop in a zone of high activation and low inhibition, while later forming cusps emerge in areas where inhibitory signals from existing cusps are weaker. Simple genetic alterations to these developmental pathways, adjusting the strength or range of activating and inhibiting signals, can produce major morphological changes. This developmental flexibility helps explain how tribosphenic features could evolve through gradual modifications of existing developmental programs, and why similar dental patterns could arise independently in different lineages. The fossil record has preserved intermediate stages in this transformation. A specimen from the Middle Jurassic deposits of China, dating to approximately 165 million years ago, exhibits a fascinating mosaic of primitive and derived dental characteristics. The molars retain 3 cusps, as in more primitive forms, but these cusps show the beginnings of triangulation. The metaconid has begun to shift lingually, though not as far as in fully developed tribosphenic molars. Most significantly, a small talonid structure appears at the rear of the lower molar, not yet developed into a full basin, but clearly prefiguring that innovation. The upper molars show a swelling on the lingual side that represents an incipient protocone, though this cusp had not yet achieved the prominence seen in later tribosphenic mammals. Such transitional fossils demonstrate that the evolution of tribosphenic molars was not a single sudden event, but a gradual process involving multiple intermediate stages, with natural selection progressively refining each component of the system. The mechanical advantages of tribosphenic molars become even clearer when we consider the broader context of early mammalian ecology and physiology. As we discussed in earlier chapters, the earliest mammals were small, probably nocturnal animals with elevated metabolic rates compared to their reptilian contemporaries. Elevated metabolism demands efficient nutrient extraction from food, which in turn requires thorough mechanical processing. Tribosphenic molars provided precisely this capability, breaking food into fine particles that could be rapidly digested and absorbed. A 2016 study by Conniff and colleagues examined physical models of teeth from 2 extinct insectivorous mammals, comparing a form with cusps arranged in a line to one with cusps in a triangle, directly paralleling the evolutionary transition we have been discussing. Researchers created replicas of the fossil teeth and tested them on artificial food materials with mechanical properties mimicking insects and other potential prey items. The experiments revealed that the triangulated arrangement required 30% less force to initiate fracture in hard materials, and reached peak force 40% more quickly than the linear arrangement. Even more significantly, the triangulated design inflicted 2 to 3 times as much damage to both hard and soft test materials, creating more numerous fractures and reducing particle size more effectively. These quantitative results led researchers to conclude that the evolution of cusp triangulation was driven primarily by selection for maximizing damage to food, with biomechanical efficiency serving as a secondary benefit. The versatility of tribosphenic molars opened new dietary opportunities for early mammals. While forms like Morganucodon were probably restricted to relatively soft-bodied insects and other easily processed prey, tribosphenic mammals could tackle harder prey items, including beetles with tough exoskeletons. The grinding capability of the talonide basin also permitted processing of food items with hard outer shells and softer interiors, efficiently extracting nutrients from resources inaccessible to mammals with simpler dentitions. This dietary expansion contributed to ecological diversification, allowing tribosphenic mammals to occupy niches unavailable to their predecessors. As tribosphenic mammals radiated into diverse ecological roles, their molars underwent further modifications and elaborations. Many lineages added a 4th major cusp to the upper molar, the hippocone, positioned on the lingual side posterior to the protocone. The addition of the hippocone created a more or less square upper molar surface, greatly expanding the grinding area. Upper teeth with this configuration are termed quadrate or quadratubercular, and they appear in numerous mammalian groups, including hedgehogs, raccoons, and many primates. Between the major cusps, small intermediate cusps called connules often developed, adding additional cutting edges and occlusal complexity. In herbivorous mammals, where grinding tough plant material became the dominant dietary requirement, tribosphenic molars evolved into highly specialized forms. Many herbivores developed lophodont teeth, where the cusps are connected by elongated ridges called lofts that run across the tooth surface. These ridges create extensive shearing edges, perfectly suited for breaking down fibrous vegetation. In the most extreme cases, such as modern elephants and some rodents, the molars become a series of parallel transverse plates, a condition called loxodont. Other herbivores evolved solenodont teeth, where the primary cusps are elongated in an anterior-posterior direction, creating crescent-shaped ridges ideal for grinding. The basic tribosphenic pattern also provided the foundation for the highly specialized carnassial teeth of carnivorous mammals. In modern dogs, cats, and their relatives, one pair of teeth in each jaw has become modified into blade-like structures optimized for slicing meat. These carnassials retain the fundamental tribosphenic architecture, but with certain components enlarged and others reduced, creating teeth that function like scissors for cutting through flesh and sinew. The upper carnassial is typically the last premolar, while the lower carnassial is the first molar, and together they form a highly effective shearing system derived from the ancestral tribosphenic design. The evolutionary success of tribosphenic molars is reflected in their taxonomic distribution. These teeth characterize an enormous clade encompassing all modern marsupials, all modern placentals, and numerous extinct relatives of both groups. The total diversity of mammals with tribosphenic molars, past and present, numbers in the tens of thousands of species, ranging from tiny shrews to enormous whales, from subterranean moles to flying bats, from grazing ungulates to predatory carnivores. This spectacular adaptive radiation was built upon the foundation of the tribosphenic molar, which provided the versatile food processing capability necessary for mammals to exploit diverse resources in varied environments. The precise timing and geographic location of tribosphenic molar origins has been a subject of intense research and debate, a topic we will explore in detail in our next chapter. What remains clear, however, is that once this dental innovation appeared, it provided such significant advantages that it rapidly became the dominant molar pattern in multiple mammalian lineages. The combination of efficient cutting, crushing, and grinding in a single tooth represented a level of functional integration rarely achieved in evolution, creating a tool that could be modified and specialized for countless dietary niches while retaining its fundamental effectiveness. The tribosphenic molar stands as testimony to evolution's capacity for innovation through the modification of existing structures. From the simple conical teeth of early synapsids, through the increasingly complex molars of cynodonts and early mammaliforms, to the sophisticated three-dimensional architecture of fully developed tribosphenic teeth, we can trace a pathway of gradual elaboration driven by the relentless pressure of natural selection, favoring more efficient food processing. Each improvement in dental function translated directly into enhanced survival and reproduction, gradually assembling the intricate system of cusps, crests, and basins that characterizes these remarkable teeth. In the chapters that follow, we will explore the geographic origins of this innovation and discover that the story is even more complex and fascinating than early researchers imagined. Chapter 5: The Southern Revolution. For more than a century and a half, the story of mammalian origins was written almost exclusively from fossils found in the northern hemisphere. The great fossil beds of Europe, North America, and Asia yielded specimen after specimen, documenting the gradual evolution of mammalian characteristics through the Mesozoic era. From these northern deposits came the first discovered Morganucodonts, the early Triconodonts, the Symmetrodonts, and eventually the earliest known tribosphenic mammals. This geographic pattern in fossil discoveries led naturally to a compelling narrative. Mammals, and specifically tribosphenic mammals, originated on the northern continents of Laurasia and only later spread to the southern landmasses of Gondwana. This northern hemisphere-centric view of mammalian evolution seemed solidly established by the mid-20th century. Paleontologists had documented a beautifully gradual sequence of dental evolution in northern fossils, showing step-by-step progression toward increasingly tribosphenic features. The oldest definitively tribosphenic mammals from northern sites dated to the early Cretaceous, appearing in deposits roughly 125 to 130 million years old. These ancient tribosphenic mammals from Mongolia, China, and other northern localities showed relatively primitive features, consistent with their interpretation as early representatives of the group. Later Cretaceous deposits from the northern continents yielded progressively more derived tribosphenic forms, eventually giving rise to the clearly recognizable ancestors of modern marsupials and placentals. The story seemed complete, coherent, and convincing. The importance of this narrative cannot be overstated. Tribosphenic molars represented one of the most crucial innovations in mammalian evolution, enabling more efficient food processing that supported higher metabolic rates and greater dietary flexibility. Understanding where and when this breakthrough occurred was fundamental to reconstructing the early history of the mammalian lineages that would ultimately dominate terrestrial ecosystems worldwide. Then, in 1997, everything changed. A team of paleontologists led by Thomas Rich and Patricia Vickers-Rich announced the discovery of a remarkable fossil from the southern coast of Victoria, Australia. The specimen came from outcrops of the Wonthaggi Formation at a site called Flat Rocks, in deposits dating to the uppermost Barremian stage of the early Cretaceous, approximately 115 to 120 million years ago. The fossil consisted of a partial lower jaw with beautifully preserved teeth, and those teeth were unmistakably tribosphenic. The animal was named Ausktribospenos nyctos, the genus name combining the Greek word for southern with a reference to tribosphenic molars, and the species name meaning of the night, acknowledging the probable nocturnal habits of this tiny mammal. What made Austrobisphoscenos so remarkable was not simply that it possessed tribosphenic molars, but rather the particular combination of features it displayed. The animal had 3 tribosphenic molars in each jaw quadrant, the same number found in early placental mammals. The transition from the premolars to the molars was gradual, with the rearmost premolar showing a molariform shape, again similar to the pattern in early placentals. Yet the lower jaw retained a distinct Mammalian groove, the trough that housed Meckel's cartilage, and clear facets where the coronoid and syrangular bones had attached. These were primitive features, expected in early mammaliaforms like Morganucodon, but supposedly absent in tribosphenic mammals. Most puzzling of all, the dentary lacked a distinct angular process, instead showing minimal inflection at its posterior margin. Rich and his colleagues proposed 3 alternative hypotheses to explain this unusual mosaic of characteristics. Perhaps Austrobisphoscenos was an early placental mammal, a eutherian that had somehow reached Australia at a very early date. Alternatively, it might belong to a sister group to the placentals, closely related but not quite a member of that clade. Or, most intriguingly, Austrobisphoscenos might represent a tribosphenic mammal entirely unrelated to the northern tribosphenic radiation, having evolved its complex molars independently on the isolated southern continent. The reaction from the paleontological community was swift and divided. Some researchers immediately questioned whether Austrobisphoscenos could truly be a placental, or even a close relative of placentals, given the primitive nature of its jaw structure. Zofia Kiel-Jaworowska and her colleagues argued that the plesiomorphic dentary indicated Austrobisphoscenos was likely a relic of a more ancient radiation, perhaps descended from primitive Symmetrodonts and unrelated to true tribosphenic mammals. However, Rich and colleagues countered that the stem placental Procanalestes also retained a Mammalian groove and facets for coronoid bones, suggesting these primitive jaw features were not necessarily incompatible with tribosphenic status. The debate quickly became heated, with fundamental questions about mammalian classification and biogeography hanging in the balance. The discovery of Austrobisphoscenos might have remained an isolated puzzle, an anomalous specimen difficult to interpret and easy to dismiss. But in the years that followed, additional tribosphenic mammals began turning up in southern hemisphere deposits, and some of them were spectacularly old. In 1999, a team led by John Flynn announced the discovery of Ambondro mahabo from Middle Jurassic rocks in the Mahajanga Basin of Madagascar. The age of these deposits was staggering, approximately 167 million years old, dating to the Bathanian stage of the Middle Jurassic. This made Ambondro significantly older than any tribosphenic mammal known from the northern hemisphere, predating northern forms by some 40 to 50 million years. The specimen consisted of a fragmentary lower jaw with a single molar, but that molar was clearly tribosphenic, possessing a well-developed talonid basin and the characteristic cusp arrangement. Flynn and his colleagues drew a bold conclusion from their discovery. If tribosphenic mammals existed in Gondwana during the Middle Jurassic, tens of millions of years before they appeared in Laurasia, then perhaps the entire group had originated in the southern hemisphere. From there, tribosphenic mammals might have spread northward during the mid-Cretaceous, when land connections or island chains made such dispersal possible. This hypothesis turned the traditional biogeographic narrative on its head, replacing a northern origin with a southern one. More southern tribosphenic mammals continued to emerge from the fossil record across multiple Gondwanan landmasses. In 2002, paleontologists described Asphaltomomylos patagonicus from Calovian deposits in the Cañadón Asfalto Basin of Argentina, dating to approximately 164 million years ago. 5 years later, in 2007, another team announced Henospherus mollis from even older Toarcian rocks at the Queso Rayado locality in Argentina, pushing the South American tribosphenic record back to roughly 180 million years ago. An unnamed tribosphenic mammal was reported from lower to middle Jurassic deposits in the Pranhita Godavari Valley of India, adding yet another southern landmass to the distribution. Additional Australian specimens were described, including Bishop's Whitmorei from sites ranging from Barremian to Albian in age, and Cryoparvis garidi from Barremian deposits at the Flat Rock site. The Gondwanan picture was further enriched by discoveries from Africa. Pre-tribosphenic or paramurin-like mammals from Morocco, including Hypomylos felizoni and Hypomylos micros, displayed some tribosphenic characteristics. Similarly, Branchotherulum tendagarense and Tendagarudotherrium ditrichi from Tanzania showed features suggesting a broader African participation in the southern mammalian radiation. These African forms added geographic breadth to the emerging pattern of Gondwanan tribosphenic evolution. By the early 2000s, it had become undeniable that tribosphenic or tribosphenic-like mammals had been widespread across Gondwana during the Jurassic and early Cretaceous. The geographic distribution spanned Australia, Madagascar, South America, India, and Africa, covering virtually all the major southern landmasses. Even more significantly, many of these southern specimens were substantially older than their northern counterparts. This dramatic temporal gap of 40 to 50 million years, combined with the wide geographic distribution, demanded explanation, and paleontologists proposed two competing hypotheses. The first hypothesis, advocated by J.C. Lowe, Richard Safeli, and Zofia Kilan-Jaworowska in a 2001 paper, proposed that mammals with tribosphenic molars were not monophyletic. According to this view, tribosphenic molars had evolved independently in two separate ancient mammalian groups with different geographic distributions. One group, which they named Australosphenida, was endemic to Gondwanan landmasses and included the southern tribosphenic mammals along with modern monotremes. The other group, named Boreosphenida, arose on the northern continents of Laurasia and included the ancestors of modern marsupials and placentals. Under this hypothesis, the sophisticated tribosphenic molar design had been invented twice by evolution through a remarkable example of convergent evolution. Luo and his colleagues presented a phylogenetic analysis of morphological characters supporting their hypothesis. They noted several detailed differences in dental structure between northern and southern tribosphenic mammals, including features of the cingulate and premolar structure. The convergent evolution hypothesis generated considerable controversy, as many researchers questioned whether such morphological complexity could evolve twice independently. An opposing hypothesis proposed that tribosphenic mammals were indeed monophyletic, having evolved only once, but that this single origin occurred in the southern hemisphere rather than the north. According to this view, tribosphenic mammals arose in Gondwana during the Jurassic period, approximately 165 million years ago, or perhaps even earlier. They subsequently dispersed to the northern continents during the early Cretaceous, when shifting continental positions and changing sea levels created dispersal opportunities. Under this scenario, the southern tribosphenic mammals represented the ancestral stock from which all later tribosphenic radiations descended, including the lineages leading to modern marsupials and placentals. This southern origin hypothesis drew support from the ages of the fossils. The oldest reliably dated tribosphenic mammals from the southern hemisphere were indeed substantially older than their northern counterparts. Henospherus from Argentina dated to the early Jurassic, approximately 180 million years ago. Ambondro from Madagascar was Middle Jurassic, around 167 million years old. In contrast, the earliest definitive tribosphenic mammals from northern deposits dated to the early Cretaceous, approximately 125 to 130 million years ago. This extraordinary temporal gap of 40 to 50 million years represented a profound challenge to the traditional view of northern origins and strongly suggested a southern origin, followed by later northward dispersal. Proponents of southern origins also noted that the southern landmasses formed a relatively interconnected supercontinent, Gondwana, during the Jurassic period. Animals could potentially disperse among South America, Africa, Madagascar, India, Antarctica, and Australia via land connections or short water gaps. This continental configuration would have facilitated the wide dispersal of tribosphenic mammals across southern landmasses that we observe in the fossil record. In contrast, dispersal from Laurasia to Gondwana would have required crossing major oceanic barriers, which seemed less plausible for small terrestrial mammals. The debate intensified with new discoveries and analyses. In 2020, Rich and an international team, including Timothy Flannery, Ann Musser, and others, described additional Australian material and provided new anatomical details about the southern tribosphenic mammals. They argued for grouping the Australian, South American, and possibly Madagascar forms into families including Bishopedidae, Tribosphenidae, and Henospheridae, interpreting these as stem therians rather than relatives of monotremes. This classification challenged the Austrospenida concept and suggested the southern tribosphenic mammals were indeed part of the lineage leading to modern therians. The critical breakthrough that would ultimately reshape the debate came with the discovery and description of upper molars attributed to southern tribosphenic mammals. For decades, previous classifications had been severely hampered by the lack of upper dental material, forcing researchers to rely almost entirely on lower jaw and tooth characters when making phylogenetic assessments. The Flannery team emphasized that this absence of information from the upper dentition had made earlier classifications inherently ambiguous. The discovery of new specimens from Australian sites that preserved upper molars with sufficient detail for meaningful comparison changed everything. These upper molars displayed prominent protocones and well-developed stylar cusp C, features characteristic of therian tribosphenic molars. The presence of these structures provided powerful evidence that the southern forms were more closely related to northern tribosphenic mammals than the convergent evolution hypothesis would predict, essentially undermining one of the key pillars of the Australosphenida concept. Recent research has tilted decisively towards supporting a southern origin for tribosphenic mammals, with subsequent dispersal to the north. A comprehensive review published in 2020 synthesized the accumulated evidence and concluded that Tribosphenidae likely arose in the southern hemisphere during the early Jurassic, approximately 50 million years before the clade's reliably dated first appearance in the northern hemisphere. The study noted that important dental modifications occurred within the southern lineage between the Jurassic and late early Cretaceous, including the development of a graduated transition between premolars and molars, the evolution of specific molar wear facets, loss of facets for postdentary bones, reduction of the Mammalian groove, and development of a true dentary angle. These progressive changes documented an evolutionary sequence occurring in southern mammals before tribosphenic forms appeared in the north. The biogeographic evidence supporting southern origins has grown stronger as our understanding of Mesozoic paleogeography has improved. During the Middle Jurassic, when the oldest tribosphenic mammals appeared in Gondwana, the southern continents were indeed well connected. South America, Africa, Madagascar, India, Antarctica, and Australia remained joined or closely adjacent, forming a vast interconnected landmass spanning the southern hemisphere. In contrast, Gondwana and Laurasia were separated by the Tethys Sea, a major oceanic barrier. Dispersal among southern landmasses would have been relatively straightforward for terrestrial organisms, while movement from south to north would have been far more challenging. However, by the early Cretaceous, when tribosphenic mammals first appeared in northern deposits, the paleogeographic situation had changed. Ongoing continental drift had begun fragmenting Gondwana, with Africa and South America separating from the other southern continents. Simultaneously, changing sea levels and shifting tectonic plates created land bridges or island chains that could have facilitated biotic interchange between north and south. India, in particular, was moving northward on its eventual collision course with Asia, potentially providing a dispersal corridor for mammals to move from Gondwana to Laurasia. The timing of these paleogeographic changes aligns well with the first appearance of tribosphenic mammals in northern fossil deposits. Despite the accumulating evidence for southern origins, the debate has not been entirely resolved. Some researchers continue to advocate for convergent evolution, pointing to detailed morphological differences between northern and southern forms. The question of monotreme relationships remains particularly contentious. If monotremes are indeed related to the southern tribosphenic mammals, as the Australosphenida hypothesis suggests, this would imply a fundamental division within mammals more ancient than the tribosphenic radiation. Alternatively, if monotremes represent a completely separate lineage that diverged before tribosphenic molars evolved, then the southern tribosphenic mammals could be stemtherians without contradicting monotreme phylogeny. The geographic origins debate illustrates how new fossil discoveries can overturn long-held scientific conclusions, and how our understanding of evolutionary history remains subject to revision as new evidence emerges. The traditional view of northern origins was based on the available evidence at the time, which consisted almost entirely of northern fossils. Once southern specimens began to surface, that view had to be reconsidered and ultimately revised. The process has been neither quick nor simple, involving decades of fossil hunting, detailed morphological analysis, phylogenetic studies, and paleobiogeographic reconstruction. Today, the weight of evidence increasingly supports the hypothesis that tribosphenic mammals originated in the southern hemisphere during the Jurassic period and later dispersed to northern continents during the Cretaceous. This conclusion transforms our understanding of mammalian biogeographic history, recognizing Gondwana rather than Laurasia as the cradle of one of the most important innovations in mammalian evolution. Yet the story is undoubtedly more complex than any simple narrative of southern origins can capture, involving multiple dispersals, regional extinctions, and parallel evolutionary experiments across both northern and southern landmasses. As we shall see in our final chapter, the evolution of tribosphenic and tribosphenic-like dental features involved convergence and parallelism even beyond the major north-south divide, revealing the extraordinary evolutionary flexibility of mammalian dental systems. The discovery of pseudo-tribosphenic mammals demonstrated that the selective pressures favoring efficient food processing could drive convergent evolution of remarkably similar dental solutions in distantly related lineages. Small body size, elevated metabolic demands, and insectivorous or omnivorous diets created common challenges for diverse Mesozoic mammalian groups. Animals facing these challenges benefited from teeth that could both cut and crush, efficiently reducing food to small particles suitable for rapid digestion. The tribosphenic design and its pseudo-tribosphenic analog represented optimal engineering solutions to these biomechanical problems for fundamental physical reasons. The pestle and mortar arrangement maximized contact area between opposing tooth surfaces during the crushing phase, distributing forces effectively and preventing fracture. The combination of tall, sharp crests for cutting and broad, flat basins for grinding allowed a single tooth to perform multiple functions, eliminating the need for specialized tooth types and conserving space in small jaws. The interlocking geometry ensured precise occlusion, preventing food from escaping between teeth and maximizing processing efficiency with each chewing cycle. But convergent evolution of tribosphenic-like features was not limited to the dramatic example of pseudo-tribosphenic molars. Even within mammals possessing conventionally oriented tribosphenic teeth, evidence suggested multiple independent acquisitions of key features. The protocone, that critical innovation defining true tribosphenic mammals, appears to have evolved at different times in different lineages. In some groups, the protocone emerged as an enlargement of the anterior portion of the lingual cingulum, gradually swelling into a distinct cusp. In others, it developed from a different starting point, perhaps as an elaboration of existing lingual structures. The end result looked the same, a prominent crushing cusp on the lingual side of the upper molar, but the developmental and evolutionary pathways differed. Similarly, the talonide basin of the lower molar showed signs of multiple independent origins. In some mammalian lineages, the talonide developed through posterior extension of the tooth crown, with new cusps appearing to border an expanding basin. In other groups, the talonide seems to have originated through modification of pre-existing posterior structures, transforming what had been a simple heel of the tooth into a functional grinding surface. Detailed studies of cusp homologies, tracing which cusps correspond between different groups, revealed complex patterns suggesting repeated parallel evolution of similar structures. Evidence for experimentation with tribosphenic-like features also appears in the African fossil record, where Jurassic and early Cretaceous mammals, such as Hypomylos, exhibited some characteristics suggestive of incipient tribosphenic organization, though they did not develop the fully realized pattern seen in later forms. Modern developmental biology has provided crucial insights into how such convergent evolution might occur at the molecular level. The development of tooth cusps is controlled by an interactive network of signaling molecules and transcription factors that regulate where and when different parts of the developing tooth form. One particularly important framework is the inhibitory cascade model, which describes how the relative expression of activators and inhibitors during tooth development influences the final number, size, and position of cusps. According to this model, cusps develop in sequence, with earlier forming cusps secreting inhibitory signals that suppress the formation of additional cusps nearby. The balance between activating signals, which promote cusp formation, and inhibiting signals, which suppress it, determines the spacing and size of cusps. A higher ratio of activator to inhibitor expression produces more numerous and larger cusps, while a lower ratio results in fewer, smaller cusps. The timing of when these signals act during tooth development further influences cusp position and arrangement, with earlier activation producing more anteriorly positioned structures. Significantly, modifications to this developmental system can produce major changes in tooth morphology through relatively simple genetic alterations. Increasing the expression of activating signals or decreasing inhibitory signals can lead to the appearance of additional cusps or the enlargement of existing ones. Shifting the timing of when these signals act can change the relative positions of cusps, potentially transforming a linear arrangement into a triangular one, or promoting the development of lingual structures like the protocone. Because the same basic developmental toolkit, the same signaling pathways and regulatory genes, operates across all mammals, similar modifications to gene expression patterns in different lineages could independently produce convergent dental morphologies. A mutation affecting the expression domain of an activating signal might promote protocone development in one lineage, while a different mutation with similar functional consequences could achieve the same result in an unrelated lineage. This developmental perspective helps explain how complex tribosphenic molars might evolve repeatedly without requiring wholesale redesign of the dental system. Rather than evolving entirely new developmental programs, the evolution of tribosphenic features could result from tweaking the parameters of existing programs, adjusting the when, where, and how much of gene expression. A mutation that slightly increased activating signals on the lingual side of the upper tooth might promote protocone development. Another mutation affecting the timing of cusp formation in the lower tooth might create space for talonid basin development by suppressing inhibitory signals that would otherwise prevent posterior expansion. Natural selection would favor these variants if they improved food processing efficiency, allowing animals to extract more nutrition from the same food or to process a wider variety of food items. Gradual refinement through accumulated small changes would progressively improve the system, ultimately producing the sophisticated tribosphenic pattern.
The recognition that tribosphenic and tribosphenic-like molars evolved multiple times profoundly impacts our understanding of Mesozoic mammalian diversity. Far from being a single lineage characterized by a unique dental innovation, tribosphenic mammals represented the convergent endpoint of multiple evolutionary experiments. Different mammalian groups, evolving under similar ecological circumstances, independently discovered similar solutions to the challenge of efficient mastication. This parallel evolution multiplied the opportunities for dietary diversification and ecological specialization, contributing to the remarkable success of mammals even before the extinction of non-avian dinosaurs. The pattern also reveals important constraints on mammalian dental evolution. While tribosphenic molars could and did evolve multiple times, the solutions arrived at were strikingly similar. The pestle and mortar arrangement of protocone and talonid basin, the combination of shearing crests and grinding surfaces, the precise occlusal relationships between upper and lower teeth, all these features appeared repeatedly because they represented optimal engineering solutions to biomechanical problems. Physical and functional constraints channeled evolution along similar pathways, limiting the range of viable designs and promoting convergence. Only certain geometric arrangements could effectively combine cutting and crushing functions. Only certain cusp positions allowed stable occlusion without tooth breakage. These constraints, rooted in the physics of materials and the mechanics of force transmission, guided independent lineages towards similar endpoints. However, not all convergence led to lasting success. Many mammalian lineages that evolved tribosphenic-like dental features eventually went extinct, their experiments in dental design ending in evolutionary dead ends. The pseudo-tribosphenic mammals, despite their sophisticated molars, did not survive beyond the Mesozoic era. Various southern tribosphenic lineages likewise disappeared, either outcompeted by northern immigrants or eliminated by environmental changes. Only certain tribosphenic lineages, specifically those ancestral to modern marsupials and placentals, persisted and radiated into the spectacular diversity we see today. The stereotyped nature of tribosphenic molars, while initially advantageous, may have ultimately constrained some lineages. Teeth designed for precise occlusion between interlocking cusps and basins require exact alignment and spacing. Any disruption to this precise fit reduces chewing efficiency. This constraint may have limited the evolutionary flexibility of tribosphenic mammals, making it difficult to modify tooth proportions or spacing without compromising function. Lineages that evolved highly uniform, precisely fitting tribosphenic molars gained immediate benefits in food processing, but potentially reduced their capacity for future evolutionary modification. In contrast, mammals with less specialized dentitions retained greater evolutionary plasticity. Their teeth, lacking the intricate three-dimensional occlusal relationships of tribosphenic molars, could be more easily modified without catastrophic loss of function. This may explain why some non-tribosphenic mammalian lineages, such as the multituberculates with their parallel rows of cusps, persisted successfully throughout the Mesozoic and well into the Cenozoic, despite lacking tribosphenic molars.
Different dental designs represented different evolutionary trade-offs between immediate functional efficiency and long-term adaptability. The ultimate success of certain tribosphenic lineages, specifically those leading to modern marsupials and placentals, may reflect subtle advantages in dental function, jaw mechanics, or other aspects of biology that are difficult to discern from fossils alone. Perhaps these successful forms had evolved more efficient chewing cycles, or their teeth generated more favorable mechanical advantages during food processing. Alternatively, success may have resulted from factors unrelated to dentition, such as reproductive strategies, metabolic efficiency, or behavioral flexibility. The fossil record preserves teeth exceptionally well, but leaves many other aspects of biology unrecorded, limiting our ability to fully explain differential success among competing lineages. What remains clear is that the evolution of tribosphenic and tribosphenic-like molars represents one of the most spectacular examples of convergent evolution in vertebrate history. Multiple mammalian lineages, evolving independently, developed strikingly similar solutions to the challenge of efficient food processing. This convergence was made possible by the lability of mammalian dental development, the power of natural selection acting on small-bodied animals with high metabolic demands, and the physical constraints that channeled evolution toward optimal biomechanical designs. The Mesozoic era was a time of extraordinary experimentation in mammalian dental evolution, with numerous lineages independently discovering the advantages of cutting and crushing functions in a single tooth. The recognition of convergent evolution in tribosphenic mammals transforms our understanding of mammalian origins from a simple linear narrative to a complex story of parallel radiations, multiple experiments, and ultimate winnowing. The mammals we see around us today, from shrews to whales, from kangaroos to elephants, are the descendants of just one successful tribosphenic lineage out of many that evolved during the Mesozoic. Other lineages, equally sophisticated in their dental adaptations, equally successful for millions of years, vanished without leaving living descendants. Their fossils remind us that evolution is not a purposeful march toward predetermined outcomes, but an opportunistic exploration of possibilities, with success and failure determined by the unpredictable contingencies of environmental change, competition, and chance. The tribosphenic molar, invented multiple times by evolution's creative force, stands as a testament both to the power of natural selection to find optimal solutions and to the ultimate fragility of even the most successful adaptations in the face of changing circumstances. Our journey through deep time has revealed how mammals acquired the defining features that enabled their eventual dominance. From Morganucodon's dual jaw joint to the transformation of those very bones into exquisitely sensitive hearing apparatus, from simple shearing teeth to the revolutionary tribosphenic molar, we have witnessed evolution's extraordinary capacity for innovation through modification of existing structures. Perhaps most remarkably, the Southern Hemisphere emerges not as a peripheral theater, but as the probable birthplace of one of mammalian evolution's most consequential innovations, challenging decades of Northern Hemisphere-centric thinking. These tiny Mesozoic mammals, scurrying through Jurassic undergrowth and Cretaceous forests, were evolutionary pioneers whose accumulated adaptations laid the foundation for the spectacular diversity of modern mammals. Their story reminds us that the grand transformations of life proceed through incremental changes, each small improvement preserved and refined across millions of years. Thank you for joining this exploration of our distant mammalian ancestors and the revolutionary adaptations that ultimately made our own existence possible.